Okay, let's go ahead and get started. Welcome, everyone. My name is Kristen Schaefer, and I'm Director of CHE, the Collaborative for Health and Environment. We invite you to put a note in the chat introducing yourself if you'd like to. I'm going to start us off with a few bits of housekeeping. First, CHE is a member of the EDC Strategies Partnership, which is bringing you today's webinar. The partnership members include Healthy Environment and Endocrine Disruptor Strategies, or HEEDS, which is a program of environmental health sciences. Also Commonweal's Biomonitoring Resource Center and the Health and Environment Alliance, or HEAL. Today's webinar is also brought to you in partnership with Breast Cancer Prevention Partners. If you'd like to download today's slides, you can find them on the webinar page on our website at healthandenvironment.org. We'll put that direct link into the chat in just a moment. We'll disable the chat during the presentation, but please type your questions in the Q&A box at any point, and we'll get to as many comments and questions as we can after the presentation. If you'd like to activate closed captions, you can do that by clicking the button at the bottom of your screen. This webinar is being recorded, and it's scheduled to last for about 45 minutes. So with that, I'll hand it over to today's moderator, Sarah Howard, Program Manager at HEEDS. Sarah.
Hi, everyone. Thank you, Kristen, and thanks for joining us. So February is National Cancer Prevention Month, and to honor that, we're featuring a webinar that addresses an issue important for true primary prevention, that is, identifying environmental chemicals that might contribute to the development of breast cancer. Our speaker today is Dr. Jennifer Kay, who is a research scientist at the Silent Spring Institute, where she works to identify chemicals that could affect the risk of breast cancer, figuring out how these chemicals act, and using that information to influence chemical policy. Dr. Kay received her PhD in biological engineering at MIT, where she also held a postdoc and helped found the MIT Superfund Research Program. There, she served as a communication link between MIT Superfund researchers and government agencies, communities, academics, and the general public. So with that, I will hand it over to you.
Thanks so much, Sarah, for that introduction, and thank you all for joining us today. I am excited to tell you about some of the work that we just published, where we used the key characteristics framework to identify potential breast carcinogens. So I'll start out, in case you weren't already motivated and aware, by pointing out that breast cancer is a public health crisis. Breast cancer has become the most common invasive cancer diagnosis in the US and worldwide. And in people under age 50, breast cancer in women is 6 times more prevalent than any type of cancer in men. And it's also 2.5 times more common than the next top cancer diagnosis among people under age 50, which is thyroid cancer in females. In addition, the rate of breast cancer diagnoses is rising, especially in women who are too young to start mammogram screening. Rising rates of breast cancer in younger people in particular suggests that this is not because of genetics or higher rates of early detection. It's something about our lifestyles, including environmental exposures. So it's urgent that we identify and reduce exposures that contribute to breast cancer as fast as possible. So in the study that I'm about to tell you about, we wanted to use the key characteristics framework to identify potential breast cancer risk factors. But we also wanted to demonstrate how this approach can be used by others to identify chemical risk factors for other types of human chronic diseases as well. So what is this key characteristics framework? The key characteristics is a list of the types of things that chemicals can do to cause cancer. This list was developed by a working group at the International Agency for Research on Cancer. And it helps us to understand what ways that chemicals can cause cancer so that we can identify chemicals that do these things. Now, in the past, people mostly focused on identifying chemicals that damage DNA or are genotoxic, as the primary indicator of a chemical's potential for being a carcinogen. But the key characteristics go beyond that and expand our view of other ways that chemicals can be carcinogenic. By creating this categorized list of things carcinogens do to cause cancer, chemicals can be assessed for whether they exhibit these characteristics in cell models or in short-term animal studies to help predict cancer hazards. This is a lot faster than trying to test every chemical in a standard 2-year cancer bioassay and waiting for tumors to develop. And it can help to prioritize chemicals that exhibit multiple key characteristics for additional testing in animals. Now, until now, we've mostly seen people using the key characteristics to evaluate the weight of evidence for a chemical's carcinogenicity. So basically starting with the chemical of interest and then looking to see what types of key characteristics it has. But we haven't really seen it used as the starting point to identify chemical hazards for specific diseases based on chemical screening data. Now, since we're interested in breast cancer, we focused on the key characteristic of receptor-mediated effects, and specifically effects related to estrogen and progesterone signaling, since these hormones are tightly associated with breast cancer risk. Since many chemicals that have been identified as carcinogens to date are DNA damaging agents, we also incorporated evidence for genotoxicity into building our list of breast cancer relevant chemicals. So today I'm going to tell you about first how we identified breast cancer relevant chemicals with the key characteristics framework by integrating data from animal cancer studies and in vitro molecular effects that have been measured in cell models to identify potential chemical hazards for breast cancer. I'll then tell you about how we validated this approach by demonstrating that endocrine activity and genotoxicity data can predict chemicals that are likely to increase breast cancer risk. And then finally, I'm going to give you one example of a way that our study suggests potentially more health protective approach for interpreting chemical testing results and making regulatory decisions. And this example is related to EPA's endocrine disruptor screening program for testing pesticides for endocrine related effects. So we started this project by first thinking about what do we know about where breast cancer comes from. Now, as I mentioned, many types of carcinogens can damage DNA, and this is true for breast cancer as well. And the reason the DNA damaging agents are so good at causing cancer is because DNA damage can cause mutations that allow normal cells to become cancerous. Breast cancer is also strongly modulated by hormonal activity. Increased estrogen and progesterone are known to increase breast cancer risk, as is clear from epidemiological studies showing that estrogen plus progestin hormone replacement therapy increases breast cancer risk among postmenopausal women. Now, the reason estrogen and progesterone increase breast cancer risk is because these hormones cause breast cells to divide, and too much hormone signaling can overstimulate cell division. This uncontrolled proliferation of cells is one of the defining characteristics of cancer. Now, one way that a chemical can increase estrogen signaling is by activating the estrogen receptor directly. We know that estrogen receptor activation is important for breast cancer because many breast cancer treatments work by blocking the estrogen receptor, like tamoxifen. Other breast cancer treatments work by blocking hormone production. So chemicals that cause cells to produce more hormones can likely increase breast cancer risk. The process of hormone production is called steroidogenesis. Now, we're fortunate that EPA has developed a high throughput screen that measures chemicals' ability to induce production of both estrogen and progesterone, as well as other hormones. And we were also able to use data from EPA screening for chemicals that interact with the estrogen receptor. Now, unfortunately, there are no good screens for measuring chemicals' activation of the progesterone receptors, so we weren't able to include data for that particular mechanism in this paper. So we started building this list of breast cancer relevant chemicals using databases from the US government and the International Agency for Research on Cancer to identify chemicals that induce mammary tumors in rodents. Now, people have historically relied on animal cancer studies as the primary basis for identifying potential human carcinogens, but these studies take years and millions of dollars to conduct, so not that many chemicals have been tested this way. We therefore incorporated mechanistic evidence for hormonal activities related to breast cancer using data from cell-based assays in EPA's ToxCast chemical screening. We looked for chemicals that cause cells to produce more estrogen or progesterone, like we saw on the right side of that last slide. And we also looked for chemicals that activate the estrogen receptor directly from a different set of cell-based assays. These screens for hormone production and estrogen receptor activation have been performed on about 2,000 chemicals. So, combining these different streams of evidence, we identified 920 chemicals plus ionizing radiation that we're considering breast cancer relevant exposures. And many of the chemicals on our list have more than one of these activities. Since DNA damage is another important way that chemicals can cause cancer, we also wanted to learn about whether our breast cancer relevant chemicals also damage DNA. So we used more US and international databases of tests performed in short-term animal studies and in cells to identify genotoxic agents. A couple highlights of chemicals on our list include chemicals that have all four of these activities that increase hormone production, activate the estrogen receptor, damage DNA, and cause mammary tumors. Those include several types of dyes, as well as diethylstilbestrol, which has been epidemiologically associated with breast cancer risk in humans. We also found some steroidogenic DNA damaging mammary carcinogens. Those include things like polycyclic aromatic hydrocarbons found in smoke, several types of pesticides, as well as other dyes. And of course, we identified some well-known endocrine disruptors like phthalates, bisphenols, and parabens. Now, after compiling this list of chemicals with breast cancer relevant activities, we then wanted to confirm that this mechanistic data we were using are actually reflective of breast cancer risk. So first, we compared mammary carcinogens to all of the chemicals that were tested for these mechanistic effects. So what we're showing here is the proportion of mammary carcinogens that tested positive for the mechanistic activity against the proportion of all of the chemicals that tested positive in that assay. We found that mammary carcinogens are significantly overrepresented for being able to cause cells to produce more estrogen and progesterone. And we refer to that overrepresentation of this mechanistic effect as enrichment. So we found that mammary carcinogens are enriched for increasing hormone production, activating the estrogen receptor, having any of these endocrine disrupting activities, being genotoxic, and being endocrine disrupting and genotoxic. We then went a step further and wanted to determine whether the types of endocrine activities we looked at were predictive specifically of whether or not a chemical was likely to induce mammary tumors in animals. So we compared chemicals that induce mammary tumors, or mammary carcinogens, versus chemicals that have been tested in a cancer bioassay and did not induce mammary tumors. Again, we found that mammary carcinogens are significantly enriched for being able to increase estrogen and progesterone production, activate the estrogen receptor, have any of these endocrine disrupting activities, be genotoxic, and be both endocrine disrupting and genotoxic. Now, the data that we used for measuring activation of the estrogen receptor and some of the data that we used for identifying chemicals that increase hormone production measured an effect size. So we were able to stratify these chemicals according to their magnitudes of activity. So chemicals with a high top EDC score were in the top 25% of estrogen or progesterone synthesis inducers, or they exceeded a certain threshold for activity at the estrogen receptor. A borderline top EDC score means that the chemical was statistically significantly active in the assay, but didn't produce a very big effect. A top EDC score of none means that the chemical tested negative in the endocrine-related assays that it was tested in. Now, when we compare chemicals based on their magnitude of endocrine activity among mammary carcinogens versus non-mammary carcinogens, we see that mammary carcinogens are significantly more likely to be stronger endocrine disruptors, and they are significantly less likely to be non-endocrine disruptors or to test negative in these endocrine-related assays. And we can see this visually too, with the darker bars corresponding to stronger endocrine activity being much larger among mammary carcinogens compared to non-MCs. Furthermore, we found a statistically significant trend for increasing magnitude of endocrine activity among mammary carcinogens compared to non-mammary carcinogens. Since we also had information about genotoxicity for our breast cancer relevant chemicals, we further stratified chemicals according to their genotoxicity as well as their magnitude of endocrine activity. Now, when we first consider just the chemicals that are genotoxic, you can see the consequence of stronger endocrine activity even more clearly, with both high and medium strength endocrine disruptors being significantly overrepresented among mammary carcinogens compared to non-MCs. Again, we see this statistically significant trend for increasing magnitude of endocrine activity among MCs versus non-MCs. Now, when we look at the non-genotoxic chemicals, well, for one thing, there weren't that many non-genotoxic mammary carcinogens, but nevertheless, we found a statistically significant overrepresentation for chemicals that are non-genotoxic and non-endocrine disrupting to be non-mammary carcinogens. So mammary carcinogens are significantly less likely to test negative for endocrine activity and genotoxicity. And again, even though we only have 5 non-genotoxic mammary carcinogens, this trend for increasing magnitude of endocrine activity among mammary carcinogens versus non-MCs is still statistically significant. So, we've demonstrated not just that mammary carcinogens are significantly enriched for endocrine disrupting and genotoxic effects, but that stronger endocrine activity is particularly informative for carcinogenic potential. This helps to validate our hypothesis that inducing estrogen and progesterone production or activating the estrogen receptor suggests that a chemical is likely to increase breast cancer risk. And it shows that integrating this kind of high throughput chemical screening data can be effective for identifying potential hazards. So to wrap up the findings of this particular publication, we identified hundreds of chemicals that could potentially increase breast cancer risk by combining traditional cancer studies with mechanistic screening data. And we hope that others will use this same approach for other diseases of interest based on what we know about the mechanisms that are involved in those diseases. We validated our use of the key characteristics of breast cancer to identify potential breast carcinogens by showing that rodent mammary carcinogens are more likely to increase hormone synthesis, activate the estrogen receptor, and cause DNA damage compared to non-mammary carcinogens. We also showed that although endocrine activity can flag likely mammary carcinogens, a lack of activity does not indicate that the chemical is not a mammary carcinogen, because many mammary carcinogens still tested negative for these endocrine-related effects. This is actually not that surprising, though, because estrogen and progesterone production and activation of the estrogen receptor are important breast cancer relevant activities, but there are many others like inducing inflammation and activation of the progesterone receptor, but most of those mechanisms lack methods to screen chemicals for them. And finally, our study highlights ways that regulatory chemical assessments can be strengthened to better protect human health. We think the EPA can incorporate this way of thinking in how they're currently approaching screening pesticides for estrogenic and androgenic effects in the endocrine disruptor screening program. So in the fall, EPA released a prioritization framework to the Federal Register for following through on their decades-old requirement to assess pesticides for endocrine disrupting effects. And they're accepting public comments until February 26th. Now, this framework is specifically focused on estrogenic or androgenic activity. EDSP also considers thyroid hormone disruption, but that's not part of this particular prioritization framework. Now, EPA's first step in this prioritization scheme is to consider whether the pesticide has been tested in an updated animal study to measure effects on reproduction and development. Now, EPA describes this type of study as updated. That means it was updated in 1998 and specifically updated to better detect effects from endocrine disrupting chemicals. But this study guideline still does not require assessment of the mammary gland. Now, if the chemical has been tested in one of these types of studies, then EPA considers that data to be sufficient to detect any type of estrogenic or androgenic effects, and they do not plan to test or consider those chemicals further. The next step is to consider whether the chemical was tested in the estrogen and androgen receptor pathway models. This estrogen receptor model is the same one that we used to identify chemicals that activate the estrogen receptor in our study. And the EPA has an analogous model for measuring activity at the androgen receptor. The androgen receptor is activated by male sex hormones like testosterone. If the chemical has been tested in one of these models and tests positive, those chemicals are EPA's top priority, and they are currently encouraging people to submit data and comments for evidence about whether these chemicals are actually estrogenic or androgenic. If the pesticide has not been tested in the estrogen or androgen receptor pathway model, these are the chemicals the EPA has prioritized second, and they are inviting additional data and comments from the public on whether these chemicals should be assessed for estrogenic or androgenic action. And finally, EPA's last priority is to consider the chemicals that tested negative for estrogen or androgen receptor activity. And based on the pace of EDSP so far, it's unlikely that they will investigate these further. Now, we have several things that we're concerned about with this prioritization framework. And the first is that it's clear that EPA has biased this framework toward dismissing evidence for estrogenic or androgenic effects. The fact that EPA is prioritizing getting information from the public about whether evidence for activity in these ER and AR pathway models are actually reflective of estrogenic or androgenic activity shows that they anticipate these comments will indicate false positives in these ER and AR pathway models. We're concerned about this because EPA considers these estrogen and androgen receptor models to have been validated for detecting effects at the estrogen and androgen receptors. And as we just saw, activity in these the ER pathway model does suggest an increased likelihood that the chemical will increase cancer risk. We're also concerned that EPA is not prioritizing testing chemicals that have not been tested for estrogenic or androgenic action. These chemicals have not been tested in animals or in this validated model for detecting activity at the estrogen or androgen receptor. And we think that these should be the top priority for further investigation because the least is known about them. Finally, we're concerned that EPA is missing other types of endocrine activity, like the ability for chemicals to cause cells to produce more hormones, data that they have already produced for many of these pesticides within the last 10 years. And they're also not considering other types of hormones like progesterone. They also are not prioritizing identifying chemicals that may be false negatives in those estrogen and androgen receptor pathway models. Now, as we saw it from the slides I just showed you, estrogen and progesterone production are an important way that chemicals can increase hormone disrupting activity and increase cancer risk. And that would not be picked up in this estrogen receptor model. So we're concerned that EPA is going to miss chemicals that may be endocrine disruptors, but don't act by activating the estrogen or androgen receptors. Again, these comments on this framework are open until February 26th if you would like to submit your own. Now, I just want to wrap up by giving you a little bit of a teaser of something that we have coming soon. Now, in the paper that I just presented, we identified hundreds of potential breast cancer hazards. And now we want to extend the translatability of this list of chemicals to further prioritize chemicals for reduction in research. So I'm currently working on a publication where we will compile information about the exposure sources for these chemicals. Are they found in things like consumer products, dietary sources, industrial settings, or pesticides? We'll compile information about biomonitoring and predicted intake levels, environmental emissions, and current regulations for these chemicals. We hope that providing information about exposure risks and current regulations will help people further prioritize chemicals for exposure reduction and further study, both by considering their biological evidence for hazard risk and also considering the exposure risks associated with each of these chemicals. I will wrap up there. The citation information for the publication I described today is on the left of the slide. I also want to thank our funding sources. In particular, my time was supported by charitable donations to Silent Springs Safer Chemicals program. So thank you especially to our donors. And we also had funding from the California Breast Cancer Research Program. I am happy to take questions now, and if you have questions beyond today, you can email me. So thank you again so much for your attention.
Thanks, Dr. K. So yeah, if you have questions, type them into the Q&A box, and I will read them out. And we already have a bunch, so I'm going to start reading just from the top. Okay, first question. Are mammary carcinogens here responsible for breast tumor promotion, initiation, or are they considered complete carcinogens?
That's a great question. So I want to emphasize that many of these chemicals have not been fully assessed for their carcinogenic potential. So one of the goals in writing this paper was to provide a basis for identifying cancer hazards without necessarily having to determine everything they might do to cause cancer. We know that we have strong evidence that they could be cancer risk factors because of the endocrine disrupting and genotoxic activities that they have. Now, chemicals that are endocrine disruptors are typically thought of as increasing cancer risk primarily through promotion. It is possible that they also contribute to the initiation step of cancer development. The chemicals that are both endocrine disrupting and genotoxic are more likely to be complete carcinogens.
Right, and she also asked a follow-up. Is there any consideration for future research on mixed exposures based on the mammary carcinogens determined in this study?
Yeah, so we are working on a project where we would try to model the level of breast cancer risk that would occur from co-exposure to multiple chemicals on this list. That project is underway. We also hope that by providing this list to the public, people who have specific interests in different types of co-exposures will use this information that we've published to also follow up with these chemicals to try to understand the cancer risks associated with co-exposures. So the paper that we have coming up will hopefully allow, for example, epidemiologists to select co-exposures that may be, more likely for people who use lots of cosmetics, for example, we can identify the breast cancer relevant chemicals that are in cosmetics and then use that information to do epidemiological studies for the cancer risk associated with those products.
So is the list of chemicals you identified available? Like, is it in the study?
Yeah, so thank you for asking that. I know,
Someone asked that.
yeah, so the list is available. It's in the supplemental materials. It's table S1 in the supplement of the paper. I know it's lots of people have had that question because it's not in the main text. The reason for that is it's 920 chemicals, which is a little bit long to put into the main text of a paper. So thank you for asking, and I hope you'll take a look at the list in the supplement.
And the same person asked, do you have a template available as a starting point to submit for public comment that reflects your concerns about the EPA's framework?
Yes, we are getting ready to submit our public comment from Silent Spring to the Federal Register docket by the end of the week. So when that goes live, people will be able to see exactly what we've written and can model comments of their own if you agree with the perspectives that we share.
Great. And so that'll be on the Silent Spring website or
So
what?
the comment will be made public on the Federal Register after we submit it, and then it gets posted. Thank you, Kristen, for posting that link to the docket. You can go there to see comments that have already been submitted, and also to submit your own comments. And that's where our comment will be available.
Right. Anyone could read anyone's comment, right?
Right.
Yeah. Okay. Okay, different question. What are your thoughts on how this information can be used for increasing incorporation of your findings in breast cancer screenings or breast cancer prevention?
Yeah, so this is another way that we think our study will really benefit epidemiological studies by prioritizing chemicals of interest based on their biological activities and based on what we know about exposure concerns and biomonitoring levels. I think that by understanding which breast cancer relevant chemicals are found in different sources can help guide those types of studies. Um, and, some of these chemicals may be very well suited to epidemiological studies, for example, with occupational exposures, where you can actually separate out, who is a highly exposed group versus who is an unexposed group. And that'll really enhance our ability to understand the human breast cancer risk associated with these chemicals that we've identified.
Um, okay. Next, aside from EPA's evaluation process, what can be done to remove these harmful chemicals from products, especially in the US, where more harmful chemicals seem to be prevalent?
Yeah, I think there's a lot of opportunity for advocacy around many of these chemicals. In our next publication, I hope to highlight, some of the chemicals that are the greatest concern so that, that information can be used by regulators, not just, in EPA's evaluation of pesticides, but also in considering whether chemicals should be listed under the Toxic Substances Controlled Act or regulated under the Clean Air Act or Safe Drinking Water Act. Another way that these chemicals can be, prioritized for reduction is, and, this list can be made more usable by consumers, is if these chemicals get listed on California's Prop 65 list of chemicals of concern. So California's Proposition 65 requires manufacturers to label products if they have chemicals that are known to have effects of concern on cancer or reproduction and development. So there are different mechanisms for ways that, laws can be tailored to better protect people from these chemicals. And we are also working to share this list with, for example, green product certifiers. So, for example, Clearia makes an app that allows you to identify harmful chemicals in products. So we are sharing this list of chemicals with them so that they can incorporate these chemicals into their apps, so people will be able to see if these chemicals are contained in different types of products. We're also sharing this list with groups like California's Department of Toxic Substances, DTSC. I always forget what that acronym stands for exactly. But we have shared this list also with green certifiers like ChemSec, so that we're really working hard to get this list into the hands of people who can use it to make a difference in reducing exposures.
Okay, actually, someone from Made Safe also says we're interested in incorporating the list into their certification program too. So,
Oh, fantastic. Glad to hear it. Let
someone
me
asked
know if
about
you'd like to
clear.
chat.
Someone also asked about Clearia, so I'll consider that answered. All right. Okay, do you think other key characteristics such as oxidative stress or epigenetic effects are less relevant for mammary carcinogens, or were they not included because of their general lack of tissue specificity?
Yeah, so there are many of the key characteristics are relevant to breast cancer. The reason we didn't include things like epigenetic effects and oxidative stress is because the methods there, we wanted to use mechanistic data that has been performed on, or mechanistic studies that have been performed on large numbers of chemicals in sort of a consistent, reproducible way. So there are not really, high throughput screens for measuring specific epigenetic effects that have been specifically tied to breast cancer risk. I know that there are methods for screening potential oxidative stress from chemical exposures, but those are not, incorporated systematically into large-scale chemical screening programs. And like you mentioned, some of those mechanisms are not specific to breast cancer, which is, why we added genotoxicity data on top of our list of breast cancer relevant chemicals identified from, animal cancer studies or evidence for endocrine disruption, because genotoxicity is not specific to breast cancer.
Right. Okay. All right. There's a follow-up question on an earlier question about mixtures. They are wondering, they're interested in the sources or co-exposures that you might study, like cosmetics, certain foods, or in the case of Ohioans, a set of vinyl chloride breakdown products that may be found near a certain train derailment. I think you know which train derailment.
Yeah. And that's a great question because vinyl chloride is a mammary carcinogen. So there are many types of co-exposures that, you could identify and prioritize for further study. I think that the idea of assessing pesticide residues on food crops is one way to, you know, think about co-exposures that are relevant for a large number of people and could also be, modified by regulation of pesticides. if we, to the extent that we know what the chemicals were from that particular train derailment, we could focus on, understanding what we know about what those chemicals are and whether they are present on this list or other key characteristics that they might have that would also be informative for breast cancer or other types of cancer.
All right. Someone said they're still having trouble finding the list. It's in the supplemental material. Maybe Kristen, can you help find that and put it in the Q&A?
Yeah, I think you have to scroll down to the bottom of the page on the publication, and you'll be able to see, I think there are 3 supplemental files. And one of those is the list of, thank you. Yes.
Okay. It's in the chat.
The Excel file, table S1 contains the full list of breast cancer relevant chemicals.
So there's a question about, you mentioned a couple times dyes, and I haven't heard much about dyes, and I'm wondering if that would be something you'd look into more with the next paper.
Yeah. So,
Like what kind of dyes, in clothing or food or,
yeah, so some of these dyes are used in consumer products like clothing, other types of products like toys, um, furniture, textiles. I couldn't tell you off the top of my head where each of those dyes are used. I know that the one dye that I mentioned that is steroidogenic, genotoxic, and mammary carcinogen, it's actually used in hair dyes. So, dyes are used in lots of different things. Some of them pose, very direct consumer exposure. Some of them, something that's only used to color plastic, maybe would be less likely to be ingested by someone, although there could still be incidental exposure. But when we publish this next list, all of these chemicals will be indicated as according to what we know about whether they are found in dietary sources. So some of the data sources that we're using that are FDA's list of substances added to food and also food contact materials. So for the dyes that are on this list, there'll be a way to sort of prioritize them for reduction and further study based on the types of things you might find them in, as well as their biological effects.
All right. And then final question, which is sort of another topic. I don't even know if we should bring this up, but what about estrogen replacement therapy in postmenopausal women? I mean, I don't know if you want to address that or not, but
Yeah, so hormone replacement therapy, estrogen and progesterone combined hormone replacement therapy in particular has been very clearly linked to increased breast cancer risk among postmenopausal people. Now, there are sort of hormone replacement therapies or hormonal therapies that are less hazardous, or at least as far as we know so far. So for example, topical treatments are less, hazardous than an oral formulation. I will mention that estrogen only hormone replacement therapy is epidemiologically associated with endometrial cancer. So different types of hormone replacement therapy have different levels of risk, but there are some that are less concerning than others.
great. All right. I think that's it for the questions, and that's it for our time too. So I'm going to turn it over to Kristen, and thank you very much, Jenny. That was great.
Thank you.
Great, thank you, Sarah. And thank you, Dr. K, for your excellent presentation and such important work. Thanks also to everyone for the great comments and questions. And I'll now wrap up with just a few announcements. First, a video recording will be available on CHE's website soon, and you will all receive an email with a link to that video. We encourage you to support the work of any and all of the groups involved in the EDC Strategies Partnership. You'll find links to each group on CHE's partnership page. If you appreciate these webinars, please consider a tax-deductible donation to CHE and/or any of these partners. We also invite you to join us for upcoming webinars. Next Thursday, February 22nd at 11 a.m. Pacific, 2 p.m. Eastern, we'll be hosting a conversation with Dr. David Kriebel to discuss tattoo inks, cancer, and other chronic diseases. Dr. Kriebel will explore existing evidence of the possible health effects of tattoos and the need for additional research and regulation. Simultaneous translation in Spanish will be provided for that webinar. And we'll put a link to register into the chat in just a moment. With that, we'd like to thank all of you again for joining us today. Thanks again to Dr. K for taking time to talk with us about your important work, to all of our EDC Strategies partners, and to my colleague, Dr. Rachel Massey, for her behind-the-scenes planning and coordination of this webinar. Thanks so much, everyone.